Now let's think about the elongation phase of transcription. The RNA polymerase, the core part of the enzyme, is going to start moving along the DNA template strand, and it's going to be moving in order to synthesize the mRNA 5' to 3'. So remember, the DNA and the RNA that's being built on it are going to be anti-parallel to each other. So if the RNA is oriented 5' to 3', that means the DNA has to be oriented the opposite way, 3' to 5'. The new mRNA is going to start growing. It's going to start getting longer as each individual nucleotide is added to it. So that will be threaded through a third channel in the core, separate from the channel where the template strand is, separate from the channel where the NTPs are coming in. Now, there's another structure or two inside of the core that we need to know about, and those are referred to as the zipper and the rudder. These are physical structures, and essentially what they are is regions of this protein, this enzyme, that are jutting out of the walls, if you will, and into the space of the tunnel. So imagine that you're standing in a corridor and there's this structure that is jutting out from the wall into the space of the corridor. That's what the zipper and the rudder are. They are physical structures that are pushing out of the wall of these channels and into the space of the channels themselves. The zipper and the rudder are going to help guide this process as the core moves. What the zipper does is it continually pushes the double helix of DNA apart. As the enzyme moves forward, as it moves downstream, we need something to keep the two strands of DNA apart. And that's what the zipper is doing. It is physically forcing those two strands of DNA apart, breaking the hydrogen bonds between the bases. The rudder is a little different. What the rudder is going to do is steer, just like the name suggests. It will steer the template strand in one direction, and it will steer the coding strand in the other direction. And that's what they're showing us in this image. Here we can see the DNA molecule, again in red, the core part of the RNA polymerase enzyme in green, and here's a growing RNA molecule. And for our purposes, we'll just call it RNA, because remember, all RNA is produced through transcription. Now, you can see these dark regions that are inside the core. Remember, we've sliced this three-dimensional enzyme in half so we can peek inside. So there's a tunnel in here, and you can see the NTPs are entering into that tunnel. Here's another tunnel over here, and you can see the DNA has been threaded in there. Here's another tunnel up at the top, and you can see the RNA is coming out of that tunnel. Here in light green is the rudder, and here's the zipper. So they're structures that are jutting out into the space of these tunnels. The zipper is helping to separate the two strands of DNA. The rudder is sending one of those strands in one direction and the other strand in the other direction. In other words, it's allowing for space in between the two strands so that the RNA can be made on the template strand and the coding strand is kept separate, if you will. Notice that as the core keeps traveling downstream, what has already been transcribed is heading, let me try that again. What's coming into the core part of the enzyme is being separated, and then we're traveling downstream. What is finished being transcribed is just going to come back together as a double-stranded molecule of DNA. So this enzyme is opening up only right where it's working. And the rest of the DNA then just snaps back closed again into a double-stranded molecule. NTPs are going to get added one at a time to this growing piece of RNA. This enzyme works quickly. It's going to add about 50 NTPs per second. Now, if you think about it, it's critical that RNA polymerase can work this quickly because genes can have hundreds to even thousands of bases in them. And we need to transcribe genes in real time. The reason transcription is happening at any particular moment is because the cell has a need for that protein at this moment. So this enzyme can't be working slowly because if it did, the protein would take too long to be produced. During the elongation part of transcription, all three of the channels inside this core part of RNA polymerase are full. One of the channels has the template strand in it. One of the channels has NTPs coming in. One of the channels has the growing RNA strand in it. This is a very finely tuned protein. The structure of the protein is perfect for the function that it has. And that brings us to the termination phase of transcription. There will come a point where the entire protein coding region of the gene gets transcribed. So the question is, how does the enzyme know when it's time to stop? The RNA polymerase enzyme is going to reach the end of the gene, and it will keep going, it will keep transcribing, but it will now be transcribing a regulatory part of the gene called the termination signal. So the termination signal is part of the DNA. It no longer has information in it about building a protein. That's not its purpose. Its purpose is to help stop transcription, to end transcription. The enzyme, the RNA polymerase enzyme, will transcribe the termination signal. But because of the way the bases are ordered in this termination signal, as this piece of RNA is made, it will fold back and bind to itself and create, remember, what we call a hairpin loop. The base sequence, the order of the bases in the termination signal in the DNA, will get transcribed and produce a sequence of RNA that can fold on itself. And when that hairpin forms, what that's going to do is just gum up the works. It's going to make it so that the core can no longer function correctly. It's got a big, bulky hairpin loop stuck in one of its channels, and so it dislodges itself from the DNA and transcription ends. The RNA polymerase enzyme, it doesn't know that it's gotten to the end of the protein coding region. All it knows is that it keeps going. It just keeps doing its work. It keeps using the DNA to make RNA. But the reason it stops is because we've hit the end of the protein coding sequence, and now we're hitting regulatory sequence in the DNA, the termination signal. And what they've shown us is the enzyme it's made this beautiful RNA full of protein coding, and then it's made this part right here from the termination signal that can fold back on itself and create a loop. So this doesn't fit nicely into the channel anymore, and that's going to cause the core to release from the DNA. Transcription is over now. We are no longer using the DNA. This enzyme is no longer using it. The RNA is released. Now, I mentioned earlier that I would point out to you some of the key differences in the transcription process in eukaryotic cells. In eukaryotes, there is no sigma. Instead, there are proteins called basal transcription factors that replace sigma. Remember, sigma is the regulatory subunit of the RNA polymerase enzyme. In a eukaryote, these BTFs are going to replace sigma. They're going to come in and they're going to bind to the promoter and they're going to guide the core in and help in the initiation of transcription. In eukaryotic cells, there are many basal transcription factors. It's a more complex process in eukaryotic cells. Something else to note is that the promoters in genes in eukaryotes are more diverse. So you see an array of different promoters in different genes in eukaryotic cells. Something else to know about transcription in eukaryotes is that eukaryotes have 3 different types of RNA polymerase. Prokaryotes have only 1 RNA polymerase. In this table, we can see the 3 RNA polymerase enzymes listed and what they do, what their function is. Now, we don't need to learn the numbered RNA polymerase enzymes right now. We need to know the details of transcription in bacteria. But you should understand that there are multiple polymerase enzymes in eukaryotes, and each one does a different job. One of them is making rRNA, one of them is making mRNA, one of them is making tRNA. And the last difference to know about eukaryotic cells is this. The messenger RNA that gets made in a eukaryotic cell is going to be heavily processed before it will be used for the process of translation. By processing, I mean that it's going to be spliced, it's going to have pieces taken out of it, and it's going to have other things added to it. It's going to have what's called a cap and a tail added onto it. We don't see this in bacteria. We don't see this in prokaryotic cells. There's a little bit of processing that happens in a prokaryote, but in a eukaryote, it's quite extensive.